it's Rebecca. Welcome to the Brainy Blonde podcast. You guys, do you know what's really strange? Having these two things at the same time: survivor's guilt and imposter syndrome. Survivor's guilt and the not quite understanding why the heck I got to survive a cancer that most people don't is what has me here, trying to do something of value to prove to myself that I was worth saving, which I know is crazy, but it's a driving force for me. The funny thing is, now that I'm actually doing the thing and reaching thousands of people sharing information and inspiration, I still find myself doubting who I am to be doing this work. You've all heard of imposter syndrome, right? That sneaky little voice that comes in and makes you question if you're really qualified to do what you're doing. I get that feeling in my day job, and I've been doing it for 15 years. So why does it surprise me that it's happening now while trying to do something entirely new? I bet some of you have experienced imposter syndrome too. Researcher Dr. Valerie Young says there are 5 imposter types. 1, the one we all probably know, the perfectionist. This type of imposter syndrome involves believing that unless you're absolutely perfect, you could have done better. And you feel like an imposter because perfectionist traits make you believe that you're not as good as others might think you are. The second type is the expert. The expert feels like an imposter because they don't know everything there is to know about a particular subject. They haven't mastered every step in a process. And because there's more for them to learn, they don't feel as if they've reached the rank of expert. The third is the natural genius. In this imposter syndrome type, you may feel like a fraud simply because you don't believe that you are naturally intelligent or competent. If you don't get something right the first time around, or it takes you longer to master a skill, you feel like an imposter. And then there's the fourth, the soloist. It's also possible to feel like an imposter if you had to ask for help to reach a certain level or status. Since you couldn't get there on your own, you question your competence or abilities. And the fifth is the super person. This type of imposter syndrome involves believing that you must be the hardest worker or reach the highest levels of achievement possible. And if you don't, you're a fraud. Did you hear that too? The irony is, you can feel like an imposter both because you didn't get there on your own and needed help, or because you did get there naturally and thereby you don't have the accreditation that comes from having learned your skills as a real expert. Long story short, this is all a bunch of nonsense and self-doubt that we high achievers sometimes subject ourselves to as a matter of fact. According to research by Alexandra Skolku, imposter syndrome affects at least 70% of people. So let's make a pact to just stop. Stop with the self-doubt. One of the best ways to feel better about yourself is to help others. So whether it's in your work or your free time, you can just start there. And I'm starting here. My guest today did a little bit of both. I'm going to share with you this conversation I had with someone who's truly remarkable. He's an engineer who is pioneering a new clinical trial for brain cancer that started with saving French bulldogs. Yeah, our beloved pets get brain cancer too. And now he's using that technology to save human lives. And what makes this story all the more compelling is that Jeremy Ling had all the reasons to doubt his career decision. He had a great job as a medical device expert, but he left the security of that job to do something bigger, something risky, something that didn't guarantee the same steady paycheck, but something that combined his tremendous natural love for music and gift for mastering sound technology into a career using ultrasound to destroy cancer cells. Jeremy Ling is the Chief Technology Officer for Alpheus Medical, a startup device company pioneering new novel cancer treatments. And it's supported by some of the foremost thought leaders in brain cancer, as well as the American Cancer Society and the National Brain Tumor Society. He is also a father of 4 and a dear, dear friend. I am so excited he is here today. Thank you for taking the time to talk to me. It's funny because here's the podcast, which is because I would love to talk to you about this all the time. So I'm happy to talk to you and hear what's going on. And
Likewise, yeah.
Yeah, this is so cool. Well, I guess for all of the listeners, it would be really helpful if you wanted to just start off by talking about your clinical trial and what you're up to.
Yeah, for sure. Alpheus Medical, we're working on pioneering a new type of cancer therapy that we call sonodynamic therapy. And maybe I'll hold on just going too far into the nitty-gritty details on sonodynamic therapy, but it's at a high level, it's a combination product, a drug and a device. And so we deliver this drug orally, and the drug preferentially accumulates in cancer cells. And then we come with this very specialized type of ultrasound application. It's kind of a new type of ultrasound that we're working on pioneering that activates the drug to create a hopefully a cytotoxic effect in the cancer cells and is indiscriminate or is safe for healthy tissue. And so it's a targeted therapy, if you will, in that sense. And so, we started Alpheus Medical about 5, 6 years ago. And the first stages, the first years of the program were really about early research and development of this therapy, of the technology, of the device side, especially to try to facilitate this and figure out how to facilitate it. And now over the last couple years, we've been in our early first in human clinical trials as well as safety trials in both the recurrent as well as newly diagnosed GBM patients. So early clinical phases right now, lots of planning and process as we look towards that next step, which would be a randomized controlled trial, which is part of, where you get a much better and cleaner, true scientific look at the efficacy. Right now, a lot of what we're doing is really predicated on safety. Of course, internally, we're certainly looking at how our patients doing, what is the efficacy, but it's not a large enough sample yet to apply any sort of meaningful statistics to the efficacy equation. So
Gotcha.
it's a stepping stone approach to kind of put all of the information together and really evaluate these new therapies. So I guess from that standpoint, you could say we're very much in the thick of it here, but excited to be doing the work, and we're certainly excited about the potential we think we're seeing thus far.
Amazing. Amazing. So, just because I kind of understand the scientific terminology that you're talking about, and I already know how it works, can you describe a little bit about the patient experience? What would they be doing? To
Maybe I start with the drug side, and I'll
yeah.
kind of dive in a little bit there. What's interesting is that the drug we're using is already approved and has been in use for over a decade. It's called 5-ALA. It's a amino acid precursor. It was first approved in the US in 2017. About 10 years prior to that, it was pioneered in Germany. And so
Okay.
it's this naturally occurring amino acid. Essentially, it takes advantage of cancer cells having a slightly different heme metabolic process compared to healthy cells. And this really results in ultimately that the cancers metabolize this into a byproduct called protoporphyrin that excessively builds up in these cancer cells, right? And this is being used. So the protoporphyrin I should mention is also this fluorescent compound. And so this is being used right now across the world as a surgical assistance tool. And so
Okay.
under the correct lighting conditions, it causes those cancer cells that have metabolized it and successfully built up there. It essentially causes them to glow or light up compared to the healthy tissue. And so this allows the surgeons to visualize or clearly define the margins when they're in there doing maximum safe surgical resection of the tumor. So
Is
again,
that what they use sometimes for like precision radiation kind of options too?
there's different drugs that help the targeted radiation. This is not necessarily one of
Okay.
those, but it is, similar in terms of helping, discriminate the target area. And so
Mhm.
I mention all that is just the background of the drug is that it already exists. And we are working on new indication of that drug, right? So right now it's being used to help the surgeons help identify the margins. We're using it in this new therapy called sonodynamic therapy, right? So it's a new indication of this existing drug. And so it turns out that, in addition to its ability to make the cancer cells fluoresce that help the surgeons, it also leaves these cancer cells in this very fragile state. it's called a, the drug's also called a sensitizer. And so
Oh,
essentially what we mean by that is we're making the cancer cells sensitive or fragile. And so it's a powder that gets mixed in with water. The patient drinks it in the morning when they arrive at the clinic. We're delivering this in an outpatient setting.
well,
And then the patient, just has to hang out for about 5, 6 hours while we let that drug metabolize and let that whole conversion process that I had mentioned before, convert the 5-ALA into this protoporphyrin and leaves the cancer cells in this kind of sensitized or fragile state. And so after about 5, 6 hours of the patient hanging out and just allowing that process to occur and the metabolism to occur, then we come in with our device side of it, which is this therapeutic ultrasound. And it's much different than I think probably what people are familiar in terms of diagnostic ultrasound or imaging ultrasound.
okay.
It's a special type of ultrasound that, we've really, through our research and development program, have pioneered specifically to activate the drug. And so the patient, sits in the chair and we take the specialized type of ultrasound machine and we apply it all across the patient's head. And our goal with that is, it's this high powered ultrasound and the drug is absorbed in those cancer cells. And we broadly distribute this ultrasound everywhere across the diseased hemisphere. And maybe please forgive this analogy, but it's like applying the ultrasound in this fashion is sort of like trying to punch the cancer cells when they're already down. They're already in
Yeah.
this fragile, sensitive state. And now we're coming in and hopefully delivering that knockout blow with this kind of specialized type of ultrasound energy. And so
Wow.
that takes a few hours to get through the ultrasound treatment, and that constitutes a sonodynamic therapy treatment session. So it's a full day activity. Show up in the clinic in the morning, drink the 5-LA, hang out. Eventually, we spend a couple hours applying this specialized type of ultrasound, and then we do repeat treatments of this. We're looking at, what are the part of our study is what are the intervals and how and when do we do those repeat treatments. But, I think because it's non-invasive, there's a huge benefit there. And because it's, very comfortable and well tolerated by the patients, because the drug is not cytotoxic by itself, there's lots of benefits to that. And opens up the doors um, for this regime where we're applying this on a regular basis to essentially, hopefully, completely eradicate all of the residual cancer cells.
That's just fascinating. At this point, you're treating patients, and I understand that you've established the first kind of step, which is that it's not harmful. The drug itself was already in use for over a decade, like you said. And what are you seeing in terms of how patients are? You said it's well tolerated. Are you seeing good results? Is it too soon to tell?
Yeah, I mean, it is very, as I mentioned, we don't have adequate number of patients enrolled to make
Mhm.
any sort of statistical claims. And because we're in the midst of this, I probably can't comment too
Sure.
much on what we're seeing. But I can say that, our enrollment has been really good. I think by and large, patients are well, I mean, you certainly know patients in this demographic that are dealing with GBM tend to be motivated patients, educated patients. And then I think in addition to that, the fact that it's non-invasive makes it a little easier to consider. Really, in terms of side effects, very, minimal lower risks in terms of side effects here. So, I think has a favorable profile
Yeah.
patients. And so I'd say that we've been really happy with the engagement and the interest and enthusiasm for the trial. And as I mentioned earlier, we certainly feel like we're seeing some great potential, and we're excited to keep that moving.
It's just amazing. It's incredible. So at this point, you are obviously in trial, and it's glioblastoma patients only, originally recurrent, and now you're taking initial diagnosed patients.
Yep, that is correct. Yeah.
Wow.
This whole sonodynamic therapy process can be applied, and there's lots of research that shows the drug accumulates in many, many different types of solid body tumors. And so, certainly we think and hope that if this technology continues to show merit, that it becomes more of a platform technology and can be applicable to other types of cancers as well. And certainly, we know that there's probably high demand in the GBM space in terms of finding alternative treatments. I think over the last decade, there's been a ton of progress on cancer treatments, and I think there's been great improvements in many areas. It's been a lot tougher in brain cancer, specifically high-grade brain cancer, and there hasn't been quite as much progress in this space. And so for that reason, kind of that unmet need, at least partially why it makes sense to start in this disease state with this type of new therapy.
Well, that's wonderful because, as you said, I mean, glioblastoma patients are so motivated. The standard of care hasn't changed in decades. There are some new, exciting, promising things on the landscape, but nothing like this. I mean, was sonodynamic treatment being used for something else? How did you come up with this, you and your team?
Yeah, and I wouldn't say we came up with that.
Okay.
No, it's been researched by a number of different institutions, a lot of them academic institutions around the world. It's been researched for a number of years. And, a lot of that research has been more at like the preclinical level or the cellular level or in vitro level. So like imagine taking cancer cells in a petri dish, exposing them to these different types of sonosensitizers like Valet, and then applying different type of ultrasound regimes to it. And under these controlled study conditions, a number of these different research institutions have all shown that there's a lot of potential here and that we're definitely seeing effect. But there's a big gap or a big challenge to take it out of that research environment and try to figure out how to actually apply it in the clinic to human patients. And so it's really, I think our challenge has been how to close that gap, as well as some of the initial foundational research, but it's more of how do we apply it to patients. And, because we're relying on that ultrasound as a preferred non-invasive approach to activate the drug, one of the challenges is we have these hard heads, the skull. It's very difficult to get ultrasound through the skull. And so I think part of our early challenges were figuring out how to make this new type of ultrasound and show that we can get meaningful therapeutic levels of ultrasound, deep into the brain tissue where, and be able to distribute that across the diseased hemisphere to get a very broad, diffuse treatment. We say it's the drug's job to go find the cancer cells, and then it's the ultrasound's job to, broadly provide the activation anywhere that the cells have absorbed the sensitizer.
Wow. So is this something that you're hoping, or I don't know if hoping's the right word, but that theoretically could be done instead of surgery? Or is this tends to be I mean, I know for glioblastoma, they usually say there's residual cells, whether they can see them or not. Is it the ones that you can't get to, or?
Yeah. Yep. No, correct. So really, we view this as addition to standard of care.
Okay.
And you had made the comment just a few minutes ago that, in GBM space, there hasn't been in the past 10 years plus, there hasn't been a lot of changes. And, it's funny because Dr. Roger Stoop, who developed the Stoop protocol in 2005, which is still the standard of, which is really just defining how to apply the chemo and radiation regime, right? Dr. Stoop he's one of our scientific advisors, and we've gotten to know him well. And I've been at a number of conferences where he's speaking, and he'll hammer his fist down on the podium and be like, we have to make progress. When I released the Stoop protocol in 2005, I never thought we'd be standing here nearly 20 years later that this is still the standard. Right. And so it's this frustration of, this has been his whole career too, of how do we bring better solutions to the table? And so this therapy, sonodynamic therapy, is really intended to be additive. We're not proposing to change the standard of care. We do believe that maximum safe surgical resection is always an important starting place with this disease. And then it's what do you do from there to get in front of the rest of it? And so chemoradiation is still part of the standard of care. Again, we feel like we slot in great because, we're additive. We're not taking anything away. And
Right.
we're very safe because ultrasound's non-ionizing and it's non-invasive. The drug is it's not a cytotoxic drug, it's a amino acid precursor. So there's all these benefits to the way we can slot in with standard of care and be a tool in the toolbox that is able to be reliably applied many times over a long course of time.
Yeah. Well, that gives me so much hope to hear because, I mean, from my understanding, the tumor unmethylated or methylated, and the unmethylated tumor cells are unlikely to respond to temozolomide, the chemo used for glioblastoma, in which case you are getting a cytotoxin that's unlikely to help.
Yeah,
But, it's the
right.
standard of care, understandably. But I would sure love to see what you're doing replace the standard of care in terms of that portion, especially if patients are unmethylated. I would love to see that progress
Yeah.
made.
Right, absolutely. And again, I'll say that's hopefully one of the benefits that's working our direction is that it's, in some ways, the ultrasound delivering that knockout punch that I was describing, it's more of
Yeah,
a mechanical process there. So in the sensitizer itself, the 5-ALA, regardless of methylated, unmethylated, again, hundreds of thousands of cases of this drug being used as a surgical aid. And so we know it's in all of those tumors and all those cancer cells because the surgeons can see its fluorescence, as
regardless of methylation, that it lights
regardless.
up, so to speak.
Yep, regardless of methylation. And
Wow.
we also know it's the heme biosynthesis pathway that cancer cells, that causes this whole cascade in metabolism that this drug builds up in the cancer cells, right? So yeah, so we feel like we can be a little bit, hopefully, a lot more resilient and tolerant to the molecular portion of this disease maybe isn't as important to our type of therapy.
Got it. Wow, that gives me a lot of hope. And I know this is a theoretical question, but theoretically, do you see something like this being applicable to, for instance, like metastasized breast cancer in the brain or other types of cancers down the road?
Yes. Yeah. No, certainly that's our hope.
Yeah.
This can be a platform and absolutely, I think we have to demonstrate in your initial indications, and then we have a whole research program in the background to try to help answer that question. But we are very anxious to, continue down that pathway.
Yeah, oh, I'm sure you are. And especially, and I know you can't comment too much on it, but I imagine you're seeing success and it's so exciting. I know I told you this, but I want you to tell the story about how this all got started with the French bulldogs, because I just think it's such I don't know. I mean, we all love animals, and that was such a beautiful way for this to kind of unfold. And, they talk about, animal research and you can think of it in a negative way, but this is such a beautiful story.
Yeah. Yeah. So we've been partnered up with the University of Minnesota Veterinary Hospital, and they've been conducting for about the past 15 years now, they've been conducting a really broad research study with many different arms or many different type of treatments where they're looking at different options for treating pet dogs with naturally occurring brain cancer, specifically high grade brain cancer, so GBMs.
So,
And so,
so you're saying, these are pets that are already sick, obviously.
yep,
Yeah.
exactly. And a lot of that, there are different type of orthotopic and other type of animal studies where, the researchers are working to create the tumor or place the tumor. And those are very different than naturally occurring, where it has when it's naturally occurring, it's going to be a tougher disease to fight because it's had the ability to take hold kind of within your in situ environment and immune system, et cetera. And so what we loved about the, and so as we were developing our preclinical, program and coming through our research phases and also trying to answer some of these questions about adequately getting the ultrasound through skulls, et cetera, we learned and found out that the University of Minnesota Vet Hospital had been doing this long, 15-year study, trying different types of immunotherapies, different types of chemotherapies, different other cytotoxic drugs, et cetera, to help treat these dogs, naturally occurring PBMs.
Wow.
And so, we got in contact with the lead investigator of that study at the vet hospital, and over the course of time of getting to know them and seeing if we could be involved and be a new arm of their study, we're happy that they were agreeable to that. And so, we have about 2 to 3 years of research under our belt now. And I was just at the vet hospital last week, actually, no, earlier this week on Monday, treating a French bulldog. And so initially we had kind of a range of dogs, like we weren't necessarily breed specific, and we were really kind of more in a learning phase. But then eventually, we kind of isolated the study in order to have a more homogeneous kind of controlled study. We selected one breed. And the breed we selected is French bulldogs. And the reason we selected them is because they were the poorest performers of over the 15 years that the study had been going, and all of the different types of drugs and treatments that had been applied. Basically, the French bulldogs were non-responders to everything. Nothing they did made any sort of improvement to overall survival.
Oh my gosh.
They all, continued to, decline. And it happens very fast, unfortunately, in dogs. It's average survival after diagnosis is only a few months. And so, we said this is actually a good opportunity to have a more controlled study. Let's go after the toughest of the tough. And then, we have all of the previous French bulldogs they treated as a control group that we can use versus our new French bulldogs that we'll apply sonodynamic therapy to. And so we're getting close to publishing on that study. And it's, I can just, casually say that we're really excited to publish on it because the data looks compelling, and where these dogs used to only live a couple months, we've, been able to make some really dramatic changes to that overall survival. Again, it's a small study. Certainly you can challenge the statistics behind the small study, but we do have a really strong control group from the same internal study, same center, et cetera, that we can compare against. And then the other aspect that we can do is we also, after we apply our treatment, our sonodynamic therapy, we're able to go in and get tissue samples from the tumors, and we're able to do our own histological workup that allows us to look for and understand signs of treatment effect in the tumor as a result of applying the therapy. And so there's a whole host of things that we've been able to explore and learn. I view all of these as kind of composites. It takes a number of things that need to really help point us in the right direction and help us make decisions on how to move this therapy forward. But certainly this was and has been a really meaningful part of our research program. And then
sorry, I didn't mean to interrupt. And I don't know if you showed me a video or if it just came to life, the story in my mind, but of this sweet French bulldog that could only walk in circles. And then after treatment, like running across a field to its human parents. I don't know if I was told this story or if it just made it up in my head, but it was like tugged at my heartstrings. It's just like this, like, oh my gosh, it's working,
Yeah. The University of Minnesota has had some good, press and coverage on it. CNN did a special on them. 60 Minutes did a special on a couple years ago as well. And so they have had some great sort of press on this study. And it's true, we do get to see that. You get to know these patients pretty well. And I love these animals. And so it does feel good when you see them make improvements compared to how they actually came into the study.
Well, and, I think briefly in the last episode that I did, very briefly, probably not in enough detail, the doctor explained the phases of trials and phase 0, through. And if you might want to just quickly touch on that, because I think it's helpful to understand why, you hear about these treatments and you hear a few select people getting them, and it's like, why isn't this available yet?
Yeah, right. It's a long process, right? And so, yeah, usually it starts in this preclinical phase, which is before you can apply it to humans. How have you demonstrated that it's, the safety and hopefully some type of efficacy signal in the preclinical environment? The dog study, the canine study, very much we consider part of our overall trial program, right?
Yep.
Starts there. And then as you move into first in human application, they call those phase 0 or phase 1 studies, and it's a very small number of patients, usually one or two centers, very controlled. And really at that point in time, from FDA's perspective, really what you're looking for is confirmation of safety. And so you have to build, in some cases, like a dose escalation study where you kind of start at lower levels and build your way up. And then, hopefully you can get to your target sort of, dose that you want to deliver, that to be therapeutic. And so there's these baby steps to get up to the therapeutic level, and then you maintain that therapeutic level for some small number of patients to help establish your safety profile. In the
Okay.
meantime, of course, again, from FDA's perspective, that whole study design is about safety reporting. Of course, the patients and of course the sponsors and of course the investigators are certainly hoping there's more to it than just safety. Of course, we want to make sure we're, hopefully benefiting, obviously, and improving outcomes for the patients as well. But as the study's designed, they're safety studies. And
Yep.
then it's as you move and there's various kind of sizes of safety studies, but eventually that leads into, a pivotal trial, which again starts with a smaller group, but then eventually grows into a randomized controlled trial where
Oh,
you do have to, and that's when you're now finally to the point where you're looking at efficacy, when you have that control group in place and you've randomly assigned patients into the treatment group or the control group. And that's very much depends on what you select in terms of how many patients and how long that takes. There's a lot of questions to answer in that, but it's, years and hundreds, so these are big studies now when you get to this point. And it really depends what the appropriate endpoints are to be pursuing in these studies, as well as how many patients you have in each of the arms, the treatment arm, control arm, and the power magnitude effect that you're looking to demonstrate for your endpoint. All that kind of gets fed into this statistical analysis that helps answer exactly what's needed in terms of patient enrollment to get through it. And
okay.
so I think one of the challenges or questions that I think, there's benefits to all of kind of the different stages of the studies. But I think one of the things that can be challenging for patients when you get to the randomized controlled trials is that concern or risk. Will I get put into the sham group? So I'm just going to be a control arm. I think what and we have concerns for that too. And we certainly lobby for where you can. Let's use historical controls. But most of the time, to do the right type of science and to really understand and tease out the efficacy signal and the magnitude of effect, you need to have these control groups as part of the study so that the demographics, the sites, the specific clinical sites that you're treating at, et cetera, that you have true controls locally, not just global controls from population databases. And so, I think we feel like there's still tremendous, especially with our therapy, because we're adding to the standard of care, not changing or subtracting the standard of care. And when we get to that point where we have a randomized control trial with a sham group, those patients will still be receiving the standard of care, but they may or may not be receiving this additive sonodynamic therapy. And so hopefully it can only be additive and not subtractive. We're not taking anything away from those patients is the way we, hopefully would like to approach, when we get to that point of clinical research. There are some other groups doing sonodynamic therapy. We're not the only, as I mentioned earlier, there's it's been research at the academic level, primarily at a number of institutions for many, many years. More recently, there are a small number of groups that have started moving this into clinical applications and these kind of phase one safety studies, similar to what we're doing. I would say one of the differences of what we've seen of other groups exploring sonodynamic therapy, even all the way back to all the original academic research institutions that have explored it, by and large, those groups are all kind of borrowing or repurposing existing type of ultrasound equipment and devices. And, we've taken a different approach where we've developed from the ground up kind of a whole new type of ultrasound because we ask the question, what is appropriate for maximizing sonodynamic therapy? And that led us to develop kind of a new type of ultrasound. And folks that maybe are familiar with this will be familiar with the term focused ultrasound. And by and large, that's what we're seeing kind of all of the research groups and the other groups that are moving into the clinical space with sonodynamics therapy. They're kind of using these existing focused ultrasound machines or equipment, if you will. And we use the analogy that focused ultrasound is kind of like shining a laser beam, at the brain and at the tumor area. And so it's a very small area of treatment. And so it makes it difficult to broadly treat this disease. And we know there's a diffuse disease challenge or element to it. And so we'd prefer that instead of shining a laser beam at the problem, let's shine a flashlight at the problem so that we get the broad therapy, the broad activation. And so, under that analogy, I think that's, been one of the differences and one of the big challenges of what we've had to pioneer is, again, this new type of, it's more the medical device side. It's more of what is this new type of diffuse ultrasound that's optimized to activate the sonosensitizer and the 5-ALA, if you will.
I like what you said about shining a light.
Yeah, right. I mean, that's, one of the challenges with GBM and why it comes back is because the surgical resection does well, but usually it's very challenging to get all of it. And because it can interdigitate and it's a very slippery sort of cell, the way it kind of seeds and seats in the brain, it becomes this diffuse problem. I think one of the challenges for us, again, is coming up with a new type of ultrasound therapy, not just simply reusing or trying to repurpose existing. It's like it's trying to optimize for the therapy itself. And so, I think that for us has been, obviously it's been, part of the challenge, but hopefully it also becomes a big part of the value of hopefully what this therapy will enable for the patients.
Yeah, I think I'm picturing, I think Roger Stoop banging on the podium and you're, just continuing to want to move the needle, saying this is great, but let's keep moving forward with this while continuing to improve upon it. And I think it must be really fascinating being in the med tech world because technology is evolving so quickly.
I guess that is one thing, I know, is part of these podcasts. I know you love to dive into kind of people's stories and their journeys and the spiritual side of it and the faith side of it and kind of the meaning of this, as much as just more so than just accomplishments necessarily, right? And, I can say that, starting this project, it certainly harkened some of those, emotions in me. I've had, I started this project about 6 years ago. But prior to that, I had 20 plus years in medical technology development, medical device R&D, experience in big companies, small companies, startups alike. And I had settled into a really nice role at a mid-sized corporation. I was leading their consulting practice for the med tech division and really wasn't looking for a job. And in fact, maybe the opposite, really appreciated my job compared to all the challenges, struggles, and failures that some of the companies I consult with were facing, especially the startups. But then as I got connected with Dr. Vijay Agrawal, he's our founder and CEO, and he's a practicing neurosurgeon. the University Hospital for Albert Einstein College of Medicine in New York City.
Uh-huh.
But as I got connected with VJ and just, kind of fell in love with his passion and motivation, he does these extremely complex and dangerous brain surgeries, every week on GBM patients, and he's frustrated. It's kind of like the Rogers 2 pounding on the podium. He's frustrated that he comes out of surgery and he knows that it's a pretty, higher probability than not that these patients still are going to have poor overall outcomes. And so how do you improve? We know surgical resection is part of it, but how do we move the needle? How do we improve therapy? And so just kind of, feeding off of some of his energy there, just really kind of fell in love with the passion. But then additionally, as I started to learn more about sonodynamic therapy myself, it turns out that this is really one of my sound is really one of my passions in life. I would
Ah.
say music in
Mm-hmm.
general. So live performances, musical instruments, musical equipment. I love to kind of dork out, especially on the engineering side of amplifiers and speakers and the signal integrity that it takes to move that signal from different components in the system, all that stuff, is very much one of my number one hobbies or passions in life, if you will. So the idea of essentially hearing with sound, was like this, wow, there's always the mantra that if you can align your career with your passions, you're more likely to do your best work. And so this challenge of pioneering a new type of sound system, if you will, this ultrasound type system, very much, the challenge of it very much resonated with me and very much overlaps with a lot of my passions in life, in the way that permeates through different areas of hobbies, etc.
I
So
love that so
yeah, so
much.
that
You knew I would.
yeah, well, and then additionally, as I'm still learning about sonodynamic therapy in this project and diving in deeper just to kind of help VJ with his planning phase, there was an extremely unfortunate coincidence that at that same time, I was also watching a close personal family friend who was my age with a young family get diagnosed and start her battle with this disease. And so, that's an extremely difficult reality to see. And I know all of the listeners who have had family or friends diagnosed with GBM know what I'm talking about. It sort of rips your heart out and you're really, kind of pretty much overwhelmed with concern and compassion. So, dealing with the impact and reality of all of that, I think pushed me to consider that, at some point, Dr. Agarwal came to me and said, let's start a company. Let's figure out how to make sonodynamic therapy real. And I felt strongly like I was being called to do this work. And at that point, it was, well, it seems pretty foolish, probably. Never mind that we have no money to start the company. Never mind that I have 4 young kids and need a paycheck to make ends meet. Never mind that we don't know if this will work. No one had ever successfully taken sonodynamic therapy out of the research lab and applied it to people at that point. Never mind, I, had done a lot of consulting and knew firsthand that most of these companies don't make it, they fail. Decided to leave
Gosh,
my
Jeremy.
comfortable and safe post to take this, what many maybe would consider a foolish risk to pursue this venture. Oh, but here we are 6 years later. We still don't know where it's going, but I feel privileged and happy to be trying to move things along and improve the science and do my best to be able to look at Roger Stoop and say, we're getting there. We are
Yeah,
we are going to make improvements.
yeah, you're getting there. I mean, I just get goosebumps hearing that because those are the moments when everything's already kind of working out and you're called to just take that leap of faith, and you did. And that's what I believe life's all about. So I'm so hopeful and
Yeah.
so optimistic and so excited. And I can hear your passion. And I hope you keep feeling this driven to to chase this because
Yeah. Oh,
it
thank you.
feels like the future.
Yeah. Yeah. Of course, we certainly want to be part of, improving the care. And, I think there's opportunity to do that. And there's through this, we've been exposed to many different types of other therapies. It's, not just sonodynamic therapy. I will say to the listeners and potential patients out there, hang in there. I think there is good stuff in the research and development pipeline. Yes, it does, unfortunately take a long time, many years to get it through market approval. But I think we're excited that eventually there's going to be a whole slate of new options to help really improve outcomes.
Yeah, that's what it feels like. That I really feel so much hope around what's coming down the pipe. I mean, for cancer, for GBM, and just like you said, hang in there because if we can just buy some time, more and more comes on the market and we'll get better care.
Yep, absolutely.
Thank you so much. That was amazing.
Oh, Becky, yeah, it was really nice catching up with you. And, kudos to you. Love the energy and passion and help and support that you bring to this space, which is, very much needed for these patients and family and friends that have to fight this problem.
Thank you for saying that. I do feel very much like that's my calling. It's my antidote to survivor's guilt is feeling like, okay, I'm doing something that's helpful.
Absolutely. Yeah, no, it's great work to be doing.
Thank you. And thank you for the time and the patience. And that was really fun. I don't know about you guys, but I'm feeling so inspired after talking with Jeremy. Of course, I have a vested interest in seeing advances in cancer care. And I know many of you do too. And there's so much hope. But on a deeper level, thinking about our lives, all of our lives being finite and precious, don't we just want to feel like we lived while we are here? It can be super scary to try something new, change careers, leave a relationship that's not serving you, move across the country, write a book. But if you feel called to do something you're passionate about, something that makes you feel inspired and excited about your life, I hope you do it. You never know the impact it will have on others. Your bravery might inspire someone else. You may or may not change the world or save someone's life, but one thing you can be darn sure about, you're going to save yours. I say cancer saved my life because I live every day with purpose. I live every day knowing I'm here for a reason. And I promise you, life before this was nothing like the life I'm living now. It is so much better. And I want the same for all of you.